My article is the nuclear gene indicates cold color polymorphism in the mammals. So the main significance or take-home message of this paper, I think, is that for the first time, the scientists extracted this whole DNA sequence and amplified it from an extinct Asian animal. And this gene, the study of this gene revealed possible polymorphism in cold color in the mammals, which is a feature that is almost impossible to study from only the fossil record. So for some background information, the gene that is studied in this paper is this melanocortin type 1 receptor gene. This is a G protein coupled receptor and encodes a key protein determining hair color in both human and many other modern animals. So the dysfunction of this gene might cause light colored coating in these modern animals. And another feature of this gene is that it's a very simple gene. It only contains like 1,000 base pair and no intros and single exons. This makes it a very good starting point. So the hypothesis of this whole article at first is that mutations might occur in this melanocortin type 1 receptor gene, and this might indicate cold color polymorphism in the mammals. So based on this hypothesis, the first step of this scientist they want to do is that to amplify the whole coding sequence of this gene and to identify the possible polymorphism positions in this gene. So what they did is that they used ancient bone powders from this animal, and they used a large-scale extraction to extract the DNA from these bone powders, and they used multiplex PCR to amplify the whole coding sequence of this gene. By comparing different fragments in this gene, they found that there are 3 polymorphic positions in this gene. And all these 3 polymorphic positions can result in amino acid change. So you can see here is the 3 possible amino acid change. And all the positions, that's 21, 67, and 301, are compared to the modern African elephants. And now they have these 3 possible polymorphic positions. The next step they wanted to do is to find out the alleles. So how does these mutations combine with each other? So what they did is that they used additional samples from several other Asian animals, Asian mammals, and they used single nucleotide polymorphic typing and also PCR to amplify these polymorphic positions. And by comparing these different types and identification of the homozygous individuals, they found 2 most likely alleles, which is depicted here. So this figure shows, compared with modern African elephant, the localization of these fixed and polymorphic positions in these mammals. And you can see from here, they depicted the 7 transmembrane style of this gene, and these 3 positions as possible polymorphism positions that happens in this animal. So now they have this gene and 3 polymorphic positions and 2 alleles that are most likely to be in this animal. What they're wondering next is that does these 2 alleles show functional differences? Because if these 2 alleles show a functional difference, that might indicate that these 2 alleles are actually causing different genotypes in these animals. So what they did is that since they have these coding sequences, they constructed these sequences into a vector. So these vectors contain either allele 1, allele 2, or all the other possible mutations that might happen in combinations. So they applied these vectors into a CMP accumulation assay. So the intracellular CMP level will increase as the activity of the G protein coupled receptor increases. So they transfected the cells with this vector, and